In adaptive immunity, you run the risk of generating cells that could attack your body, an autoimmune disease. This video examines clonal deletion, a way that the body reduces the risk of autoimmunity. All blood cells come from stem cells in bone marrow. Several types of more specialized stem cells develop into the various types of blood cells. Lymphoid stem cells produce billions of B and T lymphocytes. Here we consider T lymphocytes, also called T cells, because they mature in the thymus. Every one of the T cells makes copies of a membrane protein called a T cell receptor, or TCR. Every T cell randomly forms its TCR to have a unique binding site that will be complementary to one specific antigen. There are about 1 billion different T cells, so there are about 1 billion unique TCR binding sites. Since T cells randomly form TCRs, it's certain that some of them will bind to autoantigens, antigens that are naturally part of the body. The body eliminates these self-recognizing T cells and their potential descendants or clones through a process called clonal deletion. Clonal deletion involves 2 steps that can be considered as 2 questions. First question, does the TCR recognize the body's own major histocompatibility complex, or MHC protein? If the answer is no, then the T cell undergoes programmed cell death or apoptosis. T cells that don't recognize your body's MHC won't recognize your cells and so cannot defend you. If the answer is yes, the TCR does recognize the body's MHC, then the T cell receives a chemical signal to remain alive. These T cells are beneficial because they recognize your MHC and therefore can defend you. So, every T cell is faced with the question, does its TCR recognize the body's MHC? If not, the T cell undergoes apoptosis. If the TCR does recognize the MHC, then the T cell lives and faces the second question. Before we consider the second question, we must consider a unique aspect of the thymus, which is where the T cells are maturing. Body cells display MHC proteins that carry epitopes, which are portions of that particular cell's autoantigens. Thymic cells are no exception. They display their own epitopes. However, cells of the thymus synthesize epitopes that are normally seen on other types of the body's cells. Now let's look at the second question. Does the TCR bind both MHC and autoantigen? If the answer is yes, then the T cell dies by apoptosis. Because it recognizes autoantigen, this cell could trigger an autoimmune disease, and so it must die. Interestingly, a few of these self-recognizing T cells do survive as regulatory T cells, but that's the topic for an advanced lecture. If the answer is no, that is, the T cell's TCR does not recognize both MHC and autoantigen, then that T cell survives. Surviving cells can safely leave the thymus because they won't attack the body's autoantigens. The descendants of the survivors, their clones, become the center of the body's cellular adaptive immunity. In summary, clonal deletion involves deleting cells that do not recognize the body's MHC protein. Cells that do recognize MHC survive. Survivors that recognize the epitopes of autoantigens are deleted. Survivors that do not recognize epitope on MHC become the repertoire of adaptive immune T cells.